Welcome to today's lesson. Today we are looking at paper 3 2009 and let's see what you are supposed to do. In question 1 you are provided with the bones, bone K, bone L, M and N and P. Then you are told in Roman 1, identify the bones and name part of the mammalian body where each was obtained. First of all, examine the bones carefully and see which part of the body they are from. Now, in bone K, as you can see, this is the humerus. This is the humerus, and bone L, this is the scapula, and bone M, this is the femur. This is the femur. And then bone N, this is the tibia. This is the tibia. And then bone P is the ulna and radius. Now let's answer our question. Identify the bone and where it is found. Now bone K, we have said this is the humerus. The humerus, bone K. Bone K is the humerus, and it is found in the upper forelimb. Upper forelimb. While bone L, scapula. Bone N is scapula, and it is found in the shoulder region. Shoulder region. Then bone M, I've seen that is the femur, and then it is found in the upper forelimb. The upper hind limb. The arm is known as the forelimb, while the leg the hind limb. Now the N was the tibia. Now this is found in the lower hind limb. Lower hind limb. And then P is the ulna and radius, and it is found in the lower forelimb. Lower forelimb. Yeah. Then you are told, name the parts labeled 1, 2, 3, 4 and 5 from the bones. Let's see those parts. Now, as you can see, this is the number 1, number 2, number 3, number 4 and number 5. Now the first one is which it is found in the humerus, number 1. These, they are the lateral and medial condyles. The condyles, or you can simply call them condyles. And then number 2 is found in the scapula. This is the glenoid cavity. Yeah, number 3 is found in the femur, and this is the head of the femur. This is the head of the femur. Then number 4 is found also in the femur. And it is here, this one. That one is the patella, the groove of the patella groove. It is found in the knee there. And then number 5, this is the shaft. This is the shaft. Now we can label our parts. Can label the first one we have said, the other one. Now we can name our parts. The first one, you have said the condyles, or the lateral and the medial condyles. And then the second one, you have said is the glenoid cavity. The glenoid cavity. Third one is the head of the femur, the head. Then the fourth one, the patellar groove. And then last one is the shafts. Then you are told, name the bones that form a joint with bone K at the anterior and the posterior, and in each name the type of joint they form. First of all, you should be able to understand these terms, the anterior and the posterior. The earlier video we said that anterior is the upper side, while posterior, this is the lower side. Now when you have identified like that, now you look for the bone K. Bone K, this is the humerus. And then you should be able to know at the upper side which joint they form. Bone K, they join together with the scapula. The humerus at the anterior, they join with the scapula and they form a ball and socket joint. And then posterior end, they form a joint with the ulna and radius, and then they form a joint known as the hinge joint. Then the next question we are being told, state the function of the structure labeled 6 in bone P. Now you look at the bone P. This is the ulna and radius. And then part 6, number 6, this is the olecranon process. Then you should be able to know the function of the olecranon process. The function is to prevent overstretching of the forearm or the forelimb. Also it offers a surface area for muscle attachment. You can write like that. Also, it limits the movement of ulna and radius.
I hope you are enjoying the lesson. Do me a favor and subscribe. Thank you.
Now we go to question 2. You are provided with substance labeled P, Q, X, Y, and Z. P and Q are the food substances, while X is the dilute hydrochloric acid, and Y is dilute sodium hydrogen carbonate, and Z is the Benedict solution. Carry out the test to determine the food substance P and Q. Now here you should be able to know the reagents. Now, like if for example we have X, Y, and Z, that is HCl, sodium hydrogen carbonate, and the Benedict solution. You should be able to know which food substance the 3 tests. Now these tests for non-reducing sugar. Also, Benedict solution tests for reducing sugar. Now you have a clue that we are testing the non-reducing sugar and reducing sugar using food substance P and Q. Now here, because our space is very small, let's write here. Let's use this space You write now the food you are testing for, the food substance. Now and then you write the test, and then the procedure to be followed, procedure, observation, and finally the conclusion. And now here, then you rule. Then you start with your food substance. Let's start with food substance P. We test for the reducing sugars. Now here the procedure is put 2 milliliter of solution P in a test tube. Add equal amount of 2 milliliter of Benedict's solution, and then you heat to boil. Now your observation will be the color changes. The color changes to orange. Now here the reducing sugar are present. Reducing sugar are present. Then you rule. Now we are finished with the reducing sugar with food substance P. Now here you should be able to identify the marks first. When you have seen there are 12 marks and you are testing for 2 food substance, you should test one substance to food substance. Like here we are testing now. Now here we are using now. Let's go to use Q. We test also for the reducing sugar. Then we repeat our procedure. Put 2 ml of solution Q in a clean test tube, and then you add equal amount of the Benedict solution. Equal amount of the Benedict solution. Then you heat to boil. And then here the blue color of the Benedict remains blue. The blue color of Benedict's solution remains or retains color, remains blue. Now here in Q the reducing sugars are absent. Sugars are absent. Now up to here we have good 6 marks. Now because we have seen we have the 8 good marks. Now we have seen we are testing. Now we test for the non-reducing sugar using food substance Q. Now we write non-reducing sugar. Then you say put 2 milliliter of substance P in a test tube. Then you add 3 drops of dilute HCl and then you boil for 3 minutes. Boil for 3 minutes. Then after you boil, remove and cool with cold water. Then after you have cooled, you add sodium hydrogen carbonate. Add sodium hydrogen carbonate dropwise or drop by drop until fizzing stops. Then you add 2 milliliters of Benedict solution. Then you heat to boil. Now you should observe the color changes from blue, green goes there, yellow, and then to orange, the last color. Then you see none reducing sugars, they are present. Then you get your 12 good marks. Also, you can test the non-reducing sugar using P. Then you will get your good 12 marks. Then question 3, the photographs labeled W, X, Y, and Z show seedlings that were grown under different conditions. You want to examine them. Then you can examine them carefully. Then you are told, label any 3 parts of the seedling in part W. Now you should be able to know the parts of the plant. This is a bean. You should be able to label them. Then Roman 2, you are told, name the type of this was the type of germination, not generation, germination exhibited by the seedlings. Now this is the epigil germination. The epigil germination. Then you're told, give the reason for your answer. You say the cotyledon emerge above the ground. Emerges above the ground. You are told the seedlings in photographs W and X were planted at the same time. W and X you have seen were planted in the same time. Then state the condition under which the seedlings were grown. Let's see those seedlings. As you can see, this one is very tall while this one is very short and big stem. Now this was there was absence of light because it is very tall, and this one in the presence of light. Now your answers will be seedling in photograph W was planted in a dark, grown in a dark, while in X in light, was grown in light. Then when plants grow in condition named for seedlings in photograph W, they exhibit a certain phenomenon. Name the phenomenon. The phenomenon is when you see the seedlings that are planted in the dark, they exhibit a phenomenon known as the etiolation. And this helps for the faster growth to reach the sunlight. Helps for the faster growth in order to reach the sunlight. Then you are told, using observable features, state only 3 differences between W and X. The W and the X. I will say X has broad leaves, while W has small leaves. W has small leaves, and then X has big stem, while W has thin stem. Also, X is short while W is long. X is short while W is long. Then you are told, next question, seedlings in photograph Y and Z were planted at the same time but under different conditions. Explain how the response exhibited by the seedlings in photograph Z occurred. Let's see those seedlings. They are here now, Y and Z. Now we are asked this phenomenon in Z. Now this is simply the phototrophism, the auxins, the relationship auxins and the phototropism. Now you should be able to describe the process. Now here you'll say, when seedlings being subjected to unidirectional light, they cause auxins to migrate to the dark side, of the shoot tip, Therefore, higher auxin concentration on the dark side, higher auxins concentration on the dark side, stimulates, and there are many, they stimulate rapid cell elongation on the dark side than on the light side. On the dark side than on the light side. This results into bending of the shoot tip towards the light source. Thank you.